Let's look into how this assembly process takes place. We start with a sample and we'll see how we can break that sample and how we can sequence it. So there are, in this outline, we see that we can have, at first we can have a sample preparation, then we will submit our samples for sequencing, we'll do assembly, and then in the end, and that assembly will go through a finishing process and we have the final assemblies. So DNA is collected from the biological samples, which are then followed by library preps. Remember, guys, we talked about library preps. So we take those fragments, we break them, we add adapters, and we then have some quality controls. For example, if the sequencer is giving you 100 base pair reads, your fragments should be more or less equivalent to that length. So those kind of things are there in sample preparation. Here is an overview of this sample preparation. So when we take that genomic DNA, we do those random fragmentation. So we can break into longer fragments and we can break into shorter fragments. So both the processes, they have their pros and cons, advantages and disadvantages. So what we do here is, on one side we have those 270 base pair fragments, and on the other side we have longer fragments, which are 4 to 8 kb base fragments. So depending upon your question, depending upon the sensitivity of your experiment, you can vary these lengths. Now we take these short and long fragments and we do paired end sequencing, where what we do here is we take these fragments and we submit only their ends for sequencing, and then the middle portion we skip that. So we call this part as insert. So we have, for example, here short insert. On this side we see this insert has this sign that actually is indicating that this insert is really long. So we have long inserts here. Both of them are obviously paired end reads, and sequencers, they produce tens of millions of reads on both sides. So the output from sequencer consists of many billion short unordered DNA fragments reads. So sequencer get back to us with those large number of short reads. What we do to them? We can put them together, we can align those reads, and here as we see that those fragments are aligned and we see their ends are overlapping. So in this way we can have these overlaps between these reads, and where we can put them together based upon these overlaps and assemble those contexts. So the overlap relations are captured in a large assembly graph. So these here what we are seeing in this picture is these overlaps are represented in the shape of a graph. So what we do here is we take those reads and we break them into further small fragments, like we call it as k-mers. Like for example, the read is 100 base pair. We can take a segment like 10 base pairs from it. So where our k-mer size is 10, and we then compare, or we then overlap those k-mers with one another. So when we overlap those k-mers, we recognize those k-mers as nodes, and the edges between these nodes are actually representing their overlap. So in this way we come up with a graph where these nodes and edges are there. So after that graph is refined, we put those overlapping nodes together into contexts, and we can come up with an initial set of contexts, and from those contexts we can find our scaffolds. Okay, so finally the mates, and we do have some markers on those scaffolds or on those contexts. So actually there are some sequences which we call them as markers. So they guide us how do we put these scaffolds in a shape of an order. So with the help of these markers and with the help of these mates, we kind of refine and simplify our assemblies, and finally we get back to an assembled genome. So what so far we have seen here is that genome assembly process is divided into these following phases. So we do sample preps, we do some sequencing, which is done by those next generation sequencers, and then we do these assemblies, and these assemblies are finally refined.